Hi, grade 10s. Today for lesson 4, this is a really important lesson moving forward in our unit. So I thought I would do a quick video just to talk about really the main points of this. I'm not going to go through every single page, but just wanted to kind of go over some things. So you're probably noticing some stuff from last week was really reviewed, just things, organelles in the cell and things like that. Really, the new stuff is really starting in lesson 3 and onward. And this stuff is pretty important for next year if you plan to take biology. So there's that to think about. We'll also have a small Google form quiz this week. So just keep an eye out for that. So I want to start by just explaining why I've put this diagram here. So thinking about a classroom when we're in sort of our normal place, and then thinking of a really big stadium like the Rogers Centre. So thinking about if we were right in the center of either of these spaces, if we had to try to get out to like the outside of this room or this building, it would be much easier to get out if we were in the classroom setting. So we would have a really much smaller distance to actually travel to be able to leave that space and go outside. In the Rogers Center, we'd have to go pretty far to get out of there because that's a much larger distance, that's a much larger area. So cells are actually the same way. It's much better to have a smaller cell, as our example in the classroom, than it is to have a great big cell because there's lots of important stuff that needs to be able to move in and out, such as food and other things like that. So, going into the lesson here, I know there's some things that you're asked to compare. What I want to just really briefly say is just why do cells divide? So, why do they need to actually make copies of themselves? And one big way is, or reason I should say, is for reproduction, right? So, parents and forming offspring. Next year, if you choose to take biology, we talk a lot about the sexual reproduction side, but asexual reproduction, this first column, that's what we're focusing on for grade 10. So we have a process which is called mitosis, and it's really making copies of identical cells. So we're going to get into the details of that in lesson 5, but that's just like a little bit of an intro. And you'll see here human body cells. So that's what human cells use to actually reproduce. After reproduction, there comes repair. So if you damage your skin or you damage your tissue, cells need to divide to actually repair and replace those damaged areas. And finally, for growth. So all of us started off as one fertilized egg cell. And so from there, we don't just grow that one fertilized cell to be really, really big. We divide it smaller and smaller and smaller so that our whole organism can grow. So we'll talk about that a bit more after. And cells are really reliant upon something called diffusion. And diffusion is basically movement of stuff. So I mentioned here the cell also needs water and it moves by osmosis, which some of you guys will have heard about in elementary school. And we need all of this to happen. We need to get nutrients inside, we need to get water inside. Otherwise, things that are supposed to happen in our cell just won't happen. So I want to just bring your attention down here, which is like our classroom versus stadium example. So usually in class, I would actually make little model cells with cubes because cubes are things that we've seen lots of times in math and it's very familiar. It's also really easy for us to calculate volume and surface area. And as it turns out, we can use them as a really good model for what happens with cells as well. So here, if I have a really small side length, I'm going to have a pretty large area when I compare those two numbers. So if one side is 1 centimeter, I have a little 1 centimeter square on area of one side. The volume, which we know is length times width times height, that would just be 1 times 1 times 1. So when I calculate the ratio, that's a comparison. I'm comparing the 6 centimeter squared surface area with the 1 centimeter cubed volume. So this would be like in our classroom, okay? And again, we're comparing that to a cell. So we're saying a smaller side length, smaller cell, versus if we jump to 3 centimeters, we have a much larger room or cell if we had a 3 centimeter length. And so if you look at this last column is really, really what we need here. Um, Down here at the bottom, where I would have my stadium example or a really big cell, I have a 2-to-1 ratio, and that's really low. Okay. And I want to just mention why I'm going over this is because of the ratio. Okay. This has a huge effect on efficiency when we're moving things in and out. So again, I'm going to return to my example up here, although we're really talking about cells. If someone is standing at the door of your classroom and wants to give you your lunch, okay, they only have a very small distance to get to you and you'll get your lunch and you're happy. If you're standing in the middle of Rogers Center and someone outside of the building wants to give you your lunch, it's going to take them a really long time to get all the way into the cell or all the way into the middle of the stadium. So that's the same idea with cells. We need to be able to get in food and other things that the cell needs. So a smaller cell is way better than a larger cell. So I summarize that a bit for you here, which is we need to get things from one side of a cell to the other. Okay, so this is really why we can't just allow our cells to get bigger and bigger and bigger. We need them to split off and make small cells each time. So that whole process of actually splitting or dividing the cells, we call it mitosis. And we're going to talk about it a tiny bit here, but really the main focus of tomorrow is mitosis. So we have what's called the cell cycle. And I like to think of that or kind of interpret that as the cell's life cycle. So there's a few different things that happen in that cycle. It's not just dividing all of the time. Because if all you did was divide, you would really have no time to do anything else. So if, for example, you're talking about a muscle cell, if the cell is just constantly dividing, it doesn't actually give itself any chance to be a muscle cell. So that would be a big problem. So this circle that we see here, this is the cell cycle. So this is like life cycle of the cell. I mentioned here the period of time when the cell carries out normal functions and jobs. This is what we call interphase. So the cell spends approximately 90% of its life in interphase. And we're going to mention down here, interphase has 3 parts. So G1, S phase, and G2 phase. Sometimes the cell will go into this G naught phase, which I'll tell you about at the bottom as well. I want to just mention quickly this mitosis part right here, which, as I said, we're going to talk a lot more about tomorrow, and cytokinesis. Um, that's only about 10% of the cell. And these numbers are just generalizations. There's some that spend more or less time in these phases, but this is sort of like an average, okay? So I mentioned here, why should it be 90-10 or somewhere close to that? We need the cell to be able to grow and to make copies of its DNA, make more organelles if that's necessary, and also to do its specialized function, which is really like what is its job. So if it's a muscle cell, it's acting as a muscle cell. If it's a skin cell, it's doing that job, and so forth and so forth. So more about this specialized function will come in a later lesson. So just want to mention the 3 phases of interphase to start us off. G1, we have cell growth. So there is some growth that's happening. You don't want to have too small of a cell, but really what you're looking to do is make copies of the organelles. Okay, and I mentioned here not the nucleus. The nucleus is a special process, which we're going to see in mitosis. S phase is, or stands for, I should say, synthesis. And what it's synthesizing is DNA. So the original cell that you start off with, it needs to make a complete whole copy of that DNA in order to share that with the next cell. And I mentioned right here, it's sharing it with what we call the daughter cell. So daughter cell just means whatever cell, when the cell splits, rather, it's going to have sort of the original and then it will have the daughter or the offspring cell. G2, it's preparing for cell division, and the main thing it's doing there is checking the DNA. We're going to talk about a little later, if the DNA is not perfect, the cell does not want to divide. It doesn't want to risk having mistakes. And I'll just backtrack for a quick second. The daughter cell should have the exact same information as the parent cell or the original cell. If it changes over time, we have a name for that. It's called a DNA mutation. And that's essentially mistakes in the DNA. And that's really not good. We're going to talk about why, but one of the sort of big things that can happen is formation of tumors. So we'll talk about that a little later in the class. And last but not least, there is GNOT. And I mentioned here, GNOT is kind of like a little pathway off of the normal cell cycle, and it says cell cycle arrest. So what that means in biology is that that cell has essentially left the cell cycle for a short period of time. So if they're not needing to divide, they will just leave the cycle. And there are some specific examples of that. One of the big ones is actually our neurons. So the brain, the cells rather in our brain and spinal cord. So we'll talk about them a bit more later on. And then just briefly, I'll mention a couple of details, but I want you guys to read through this yourself. The rate of cell division. So for some cells, it's really, really fast. For some cells, it's more slowly. So let's think for a second about our skin cells, especially now if you get a sunburn or something like that, and your cells start to flake off, right? You start to get dry skin and those kind of fall off from the surface. So those type of cells, they have to divide really, really fast because we lose so many of them every day, even if we can't see it with the naked eye. And then the neuron that I was mentioning earlier that I would find in the brain or spinal cord or in our nerves, they basically do not divide. And we're finding new information that they might be able to divide really, really slowly in some cases, but for the most part, they're not actively dividing. So they would be a good example to enter GNOT. Now, the rest of the stuff down here, we're starting to talk now about cell division. Okay, so I mentioned from the diagram on the last page that includes mitosis and cytokinesis. We will go through all of the nitty-gritty details of these two processes in lesson 5 tomorrow. But for now, it's just to divide essentially into two phases. So mitosis being division of DNA and forming two nuclei, which is just the plural of the nucleus. And that contains four phases: prophase, metaphase, anaphase, and telophase. We often short form this to PMAT. PMAT. So you'll see that written a couple of times. And cytokinesis, it means quite literally splitting of the cytoplasm. So that's going to be like ultimately to separate into two new cells. So just to quickly mention, the DNA has already been copied, as I mentioned before mitosis starts. So that happens during interphase. And this is so the cell can check out that the DNA is all good. Okay, so I'm going to scroll up for just a quick sec. You could do the same in your notes if you like. As I mentioned, this mitosis part is like basically splitting up the cell. That's cell division. And you can see if we follow the arrows, the S phase has happened way before that. Okay, and I mentioned before about mutations. I'm talking about negative mutations, but here the cell will try to look through and just make sure everything is all good. The cell tries to fix the mistake, but if it cannot, it could enter what's called programmed cell death or apoptosis. So that's basically the cell saying, wow, this mistake is really big. I don't know what to do with it. It's better for the cell to just stop dividing and die off rather than copying that mistake many more times. And if it does not fix the mistake, it's possible that the cell loses control of cell division. And that can lead, as I mentioned before, to tumors. It could lead in extreme circumstances to cancer as well. So we'll talk more about cancer. We have a whole assignment and a whole lesson just about that. So that will come a bit later on. I want to focus just on S phase, which is the DNA synthesis phase. You may have heard that each of our cells has 46 chromosomes, and those 46 are represented by 23 from each of our parents, okay, our biological parents. So what I'm showing down here is just a way that we're going to represent them when we want to draw them. So this little line is representing a single chromosome. So before S phase, we have 46 single chromosomes. S phase, the whole job is making an exact copy of those single chromosomes. So what we see after S phase, so when we start G2 phase, is a duplicated chromosome. So this guy has been copied into this X-shaped looking thing. And so we'll have, hopefully, if everything has gone properly, 46 duplicated chromosomes. These should all be identical to the parent, right? It should be identical to what we started over here with. You'll notice too, I mentioned this little green dot in the center. We call that a centromere. And we're going to see that centromeres are really important for tomorrow, where we start moving the chromosomes around in the cell. So that's why I've labeled it here, but we're going to talk a bit more about them tomorrow. So I hope this video helps you. Like I said, this is really an intro to cell division. Lesson 5 is like the main focus of it.